Yeah, my name is Kevin Potter from North Carolina State University, and this is work that I've done with Kurt Ritters from the Southern Research Station of the U.S. Forest Service. And we're using, we've developed a national multi-scale assessment of regeneration deficit as an indicator of potential risk of forest genetic variation loss. So a quick overview of what I want to talk about here. First of all, national forest sustainability assessments call for indicators of genetic diversity. So I'll mention that briefly, talk about that. Seed zones can be useful tools for broad-scale genetic diversity assessments like this. And we propose a genetic diversity indicator that combines provisional seed zones with national scale forest inventory data. This was described in a paper published earlier this year. And our goal here is to offer insights into which species and areas of the country are at risk of losing genetic variation because of inadequate regeneration. So first of all, in terms of national assessments of forest sustainability, one of the leading frameworks for that is the Montreal Process Criteria and Indicators, which encompasses 12 countries, including all three of the major countries of North America. And these are countries that mainly have temperate and boreal forests. These countries encompass 49% of the world's forests and 33% of the world's population. And this framework was launched in 1994 following the 1992 Rio Earth Summit, and it includes 7 criteria of forest sustainability and 67 specific indicators. The U.S. Forest Service uses the Montreal Process CNI as its main forest sustainability assessment framework, and it publishes regular reports to highlight the state of U.S. forests and tracks the trends in those 67 indicators. So the first criterion of the 7 is conservation of biological diversity. It includes 9 specific indicators, 3 each of ecosystem diversity, species diversity, and genetic diversity. With genetic diversity, these genetic diversity indicators are fairly recent additions and have not been as well developed as these others. And the work we're focusing on addresses this first indicator here: number and geographic distribution of forest-associated species at risk of losing genetic variation and locally adapted genotypes. So we're interested in addressing that for tree species specifically. So our proposed indicator quantifies tree species regeneration for about 280 tree species across the United States from forest inventory data within provisional seed zones. So I'll talk about both of these things separately, starting with provisional seed zones. So what are seed zones? Seed zones are areas in which plant materials can be transferred with little risk of being poorly adapted to their new location. They are essentially units of adaptive similarity within species. Empirical seed zones have been developed for some commercially important tree species, especially in the western part of North America. British Columbia, for example, has been a leader in this, and these tend to be based on adaptive genetic variation data from common garden experiments. So that's great for commercially important species, but in the United States, the continental United States, we have more than 400 tree species, and we want to get a sense across all of those how they're doing in terms of their adaptive variation and genetic diversity. Little, if any, information on adaptive genetic variation is available for most of those. Collecting that takes a lot of time and money, as you would expect, and time is of the essence, as several of these species are at risk from pests and pathogens and climate change, among other things. So one option is to use provisional seed zones. These are based on environmental information independent of genetics, with the assumption being that adaptive variation within species is associated with the conditions, the environmental conditions that define those zones. And we can define seed zones quickly and for many species. This is an example that was published recently at the county level based on climatic factors for the eastern United States. So we're, because we want to include the entirety of the continental United States, we are using national provisional seed zones based on climatic similarity, published by Bauer et al. A few years ago. And these seed zones are based on the intersection of minimum winter temperature with aridity across the continental United States. When they did that, they came up with 64 provisional seed zones. And then they recommend overlaying those seed zones with ecoregion treatments. In this case, the Forest Service provinces we've used to capture other conditions such as adaptive factors and that sort of thing. And in their study, they found that these seed zones were nearly as good as empirical ones for accounting for adaptive variation in native plants in the Pacific Northwest. So we ended up with 474 of these seed zones, provisional seed zones, when we overlaid the climactic zones with the ecoregions. So the 474, I should say, that contain forest and are at least 100,000 hectares, the average size of these being about 1.6 million hectares. This is an example of how those would intersect with a species, a forestry species. This is the Longleaf Pine distribution across the southern United States. It occurs in 9 provisional seed zones in which there are at least 5 forest inventory and analysis plots. So that's the first part of our indicator. The second part is getting a sense of regeneration using forest inventory data. And that comes that information comes from the Forest Inventory and Analysis Program of the U.S. Forest Service, which is this amazing program that encompasses about 140,000 forested plots nationally. These are all fixed area plots, identical design, measured 5 to 7 years in the east, 10 years in the west. And it has this really nifty statistical design that allows for aerial and tree count estimates because the plots are spatially unbiased. So the FIA program divides the country up into these hexagons of approximately 2,400 hectares, and then locates or selects a point at random within those. And if there's forest at that plot, that is the plot that gets sampled. So because of this design, each tree and plot condition represents a larger population or area. So you can make an estimate of the number of loblolly pine trees in North Carolina, for example. So our regeneration indicator is the percent of estimated stems across plots that are seedlings or saplings, that is small stems for each species relative to all of the trees within that species. So here we're using the FIA design to estimate the total number of large and small stems, because again, each plot represents a larger area, with fewer smaller stems suggesting insufficient recruitment for that species. For consistency, we defined a small stem for a species as being those that are less than 20% of the 99.9 percentile of the maximum diameter of that species, up to 12.7 centimeters DBH. And we do that to be able to compare regeneration across species with varying size distributions. So we want to make sure that we're capturing insufficient regeneration for small species like dogwood or redbud, as well as oaks and the like. So what we have here, in other words, is, or what our indicator is here essentially is if fewer than 75% of all of the stems of a species are small, then we have unsustainable regeneration and regeneration deficit. And that's calculated across a species range and within each of the seed zones that has at least 5 FIA plots in it. So And this is based on the idea of stand level balance across large scales. So aggregated at a landscape scale, even uneven stands are expected to have this negative exponential or reverse J diameter distribution. So if we have our 20% of max diameter here, if fewer than 75% of the stems are on this side of that line, then we have unsustainable regeneration, versus if we have more than 75%, then we have sustainable regeneration. So some examples.
Kevin, that was 10 minutes, just your.
Okay, great, I'm on time. So some examples here, we look at sycamore, 80% approximately of its stems across its range are small, so it's not at risk range wide, so it clears that 75% threshold, regeneration deficit threshold. However, in 18 of the 34 seed zones in which sycamore occurs, it does not clear that threshold. So if you see in New York and parts of the upper Midwest, there is sort of insufficient regeneration there that would cause us some concern about the genetic variability of that species over the long term. Table Mountain Pine, another example here, across its range, 61% of the trees stems are small, so it's at risk range wide, and in none of its seed zones does it clear that threshold. So in every one of these seed zones, we would have concerns about the regeneration of Table Mountain Pine and how it affects the genetic variation of the species over the long term. An example from the West Coast, red elder here, about 71% small stems at risk range wide, although it's kind of on the border, with 14 of the 21 seed zones not clearing that threshold and being places where we would be concerned. So of our 280 tree species that we looked at, 46 or 16% of those had regeneration deficit across their ranges. This is incorrect, it should be 75% here. Some examples are giant redwood and bishop pine in California, which had no stems, small stems at all, versus, as well as some western oaks, Table Mountain Pine, Osage orange, bald cypress. Some tree species at risk across the majority of their seed zones. There are 39 of those, so those are of concern. Some of those overlap with the ones we just looked at, but then there's some others here as well that may clear that threshold across many of their, or across the entirety of their range, but have the majority of their seed zones with issues. When we stack all these on top of each other and get geographic distribution of tree species at risk, we can get the number by seed zone, and we can see the upper Midwest and the South are the places where we have the most species that are not clearing that threshold. And then if we look at just the percent of species within those seed zones that are not clearing that threshold, we get a different picture because there's greater species diversity in the East. Parts of the Southwest and California, nearly all of the species are having regeneration issues, which is problematic, of course. So some caveats and conclusions. These provisional seed zones are far from perfect. We assume species are always well adapted to their current environmental conditions under this framework. And we assume adaptive variation is partitioned similarly across species. So those are probably not great assumptions, but it's, you know, this is what we have to work with given the number of species we're looking at. So some information, though imperfect, is better than none when we need to assess sustainability relatively rapidly across many species. And we make the case that provisional seed zones combined with national inventory data are useful for sustainability assessment and conservation decision. So I appreciate folks who have supported us with funding, the work of the FIA field crews, and the like. And if I have time for question, I will be happy to answer.
Thank you. Good morning, everybody. Yeah, this is regards a Longleaf Pine provenance test that was established in Virginia, and it was established by the state. Jerry Creighton was leading that effort, and my other co-authors are Christopher Mayer and John Butner from the U.S. Forest Service, and Gary Holly, and I did that wrong, and Paul Scherberg from University of Vermont, and Paul is also from the U.S. Forest Service. Okay, well, they originally there were, well, the issue is there were only approximately 200 mature native longleaf pine trees remaining in the state of Virginia from a population that was in the hundreds of millions, according to Jerry. So should these trees be conserved and germplasm be preserved via planting local seed sources? Are they different enough that planting, regeneration, forest restoration needs to use those seed sources? So what I have here, we used 8 seed sources, and these numbers are their minimum 30-year mean minimum temperature for the year. And so they were collected in Virginia, North Carolina orchard. I don't know exactly where these were collected, so I don't have a temperature associated with that. Another site in North Carolina, South Carolina, Alabama, Georgia, Florida, and Mississippi. So a very wide range of sources planted near the edge of the range of longleaf pine. And longleaf pine is an interesting species. It's very valuable. So when European settlers came in, they exploited it extensively for naval stores, and because this wood is so valuable, it's straight and it's high density. And now the extent of longleaf pine is 5% of what it was previous to Europeans arriving. So the design is it was planted on 3 sites up near the Richmond area, 2 blocks per site, 25 trees per block, 9 trees per provenance per block were sampled for carbon isotope discrimination. And then a much smaller sample, 1 tree per provenance per block, was sampled for cold tolerance on 4 dates. So just quickly getting into it, looking at the height growth over time of the different provenances, the 8, not a tremendous amount of variation here. It is statistically significant, but Virginia isn't always the leader, but it's usually near the top, but the differences aren't really tremendous. And that's over 10 years from the first measured at 5 years. Survival was more variable, and Virginia had the highest survival, and then it was decreasing in with the other provenances, and I'll show you in a minute that an interesting relationship that seems to explain that. And this is volume, so it's basically in a volume index. It's taking PBH times height squared times using an estimate of 440, what do I got here? 200 trees per hectare to give this volume estimate. And see, you can see here there's quite a variation with the different provenances, with Virginia being the highest. The second highest comes from that North Carolina seed orchard. But this kind of really struck me because I just did this the other day, and I had to do it a few times because I actually didn't believe it. I thought I must be doing something wrong, but if you look at 10-year survival and then you plot that versus the 30-year minimum temperature, it's pretty amazing because survival decreased linearly at a R² of 0.7 with temperature. So the warmer the temperatures that the seed sources came from that area, the less the more mortality there was. And so I thought this was wow, this is great. I'm glad I finally looked at that. We actually, so that indicates there's differences in cold tolerance, but we actually went with the University of Vermont, Butner and Hawley and Schauberg from up in Vermont, do a lot of electrolyte leakage measurements of tissue, and this actually is TM is over here on the y-axis, and that indicates the 50% where there's 50% damage in the foliage, and I really don't have time to get into how that's done, but basically earlier in the year they were more cold hardy, and they increased over time as the year went by, and actually none of these are there significant differences. But the one caveat for this is there was very small sample size for this, so we only had one tree per provenance per block across the sites. So doing this doesn't indicates there, with that sample size, that there isn't really very strong variation. There isn't variation among them, but obviously there was variation in cold tolerance from that relationship between survival and minimum temperature. Also looked at water use efficiency using carbon isotope discrimination and got some very interesting results. The Virginia seed sources have the lowest discrimination on each site by far in most cases. And this is all statistically significant. I'm trying to understand why, and remember, I don't know if I said that, most of the other seed lots come from collections of 50 trees from the different areas, and these only come from 10 trees in a very small area, the Virginia ones. So maybe there was some type of inbreeding. I don't have information on what kind of inbreeding there may have been observed in the greenhouses, because all this seed was provided by a man named Wayne Bell, who had a seed company. He's retired now, and he provided seed lots with at least 50 trees collected to provide the seeds. So that's for whatever reason, that's a stunning difference. But look how I have the scale. I have it from going from 20 to 22. So the variation isn't that, this is not very different than that, but in fact, I've done work where even those small amounts of differences in discrimination in black spruce provenance tests, progeny tests, related to growth variation. So the major results were that seed sources only had minor differences in height and diameter growth, but the Virginia sources had the highest survival. Survival was correlated to minimum temperature of provenance location. No differences in cold hardiness using electrolyte leakage, but Virginia had, with these, once again, caveat of 10 trees from a very small area, from just a few hectares, had the highest water use efficiency, the lowest discrimination on all the sites. So my conclusions are that the Virginia seed source had the highest productivity and appears to be unique with respect to water use efficiency. So the matured longleaf pine trees in Virginia really need to be protected, and the progeny from those used in restoration. However, since there's only 200 trees left in the state of Virginia, seed from other provenances with high productivity really need to be used, probably the best in a blend of the seed sources to provide sufficient genetic diversity, which is certainly lacking, most likely lacking. We don't have all that, we don't have a particular study on that within the state of Virginia, which only has 200 trees left. And I think that's the end.
Sergio Simmental cannot participate, and so I have to present. I would like to present this this presentation about genome-wide scan of detecting adaptation to local environment in Populus tremoloides, a skillstone tree species in North America. A little introduction. Forest regions are, it's an area with trees can be moved with little risk of maladaptation because of the low environmental variation. Science trees may become maladapted under climate change. Understanding current patterns of adaptive variation in species has become crucial to enlighten and guide climate resilient forestations. Well, Populus tremoloides is very common in North America and has a distribution about 6,000 kilometers from north to south. And also there are many little population in Mexico, but without informations. Well, the study aim was a genome-wide association study. And with this study, we located, delimited, and mapped Mexican populations of Populus tremoloides in the recent climate conditions. In the future, as for the, some forecast with some climate change. We expect in this study association between Mexican Populus tremoloides proveniences and local climate, soil, geographical, and topographical factors. And we used 1.5 million unfiltered SNPs from 96 populations, in total 960 samples from all parts of Mexico. In the left side, we can see a little map of this sample sites. We can see in Baja California, small populations. The most population we can find in the Sierra Madre Occidental, and also some populations in the Sierra Madre Oriental. That's here. And then we can have some populations too near to Mexico City. Well, so we filtered this 1.1.5 million SNPs with some filters, with MEC, with MAF, with MAS, and we found filtered 33,000 filter SNPs that were present in all of these filters, and 32 putative top adaptive SNPs. We have much more adaptive SNPs, but 32 very strong signals, with very strong signals. Well, then some words about the statistics. Then we selected non-collinear environmental variables and made a correlation to the relative frequency of these 32 outlier SNPs. And using 6 machine learning models. The first, the linear regression, random forest, neutral network, model avenge, neutral network, multi-layer perceptration, and Bayesian regulated neural networks. Well, And then with this information, we modeled the distribution of the species and their provenances using the random forest classification to calculate the map and delineate each definite provenance. So, well, first we find 970 populations. That's an estimation with our models. And in Mexico, we have so more or less 1,000 hectares, not more, of populous Tremorides, with a probability of presence of 99%. The red points, you can see this stands, but it's a model. So then we find several significant correlations with climate and soil variables with our SNPs, and we find up to 0.42 R square. Here can we see, for example, with the summer precipitation, the S-day, the minimum temperature in the coldest month, and in the precipitation in the vegetation time. And here we can see the different models. And most important is the RMSE, this arrow, that's the frequency of the SNPs. And we can say there are strong correlations with the environmental variables. Here is a pre-result with the summer precipitation. We found really two different provenances in red and in blue. But the most common variant is in the Sierra Madre Occidental, that we can see with the red color. So the discussion and conclusion is very small. Our provenance modeling could be helped to understand local adaptation in populous Tremorides in Mexico. Well, this was a funding from CONACYT together with the funds of Quebec. And in this study, we have 3 students that help us, also Sergio Simmental. And well, and so we appreciate the assistance of numerous field techniques that help us in this study. Many thanks. That's all.
Hello, good afternoon, everyone. Thanks to be here and listen to my presentation. Today I'm going to present you the topic field trials of growth and stomata density of Mexican pinus species and their hybrids. This investigation can be made thanks to the Juárez University of the state of Durango, the Science and Technology Council of Durango State too, and of course to the help of many teachers and researchers for different schools of Durango City in Mexico. So today we are going to talk about natural hybridization, and natural hybridization is one of the most important evolutionary forces in nature. Approximately like around 80% of the species that we know nowadays are made thanks to this force. And in plants, it's a common phenomenon. Actually, more than 25% of the plants hybridize naturally. In Mexico, we have different cases of hybridization between different important pine species. Some of them are Pinus ocarpa with Pinus caribea, Pinus ocarpa with Pinus pinglei, Pinus engelmani with Pinus arizonica, Pinus ingelmani with Pinus durangensis. These last two are some of the crosses that we are going to see on this presentation. And finally, we have Pinus Luz Maria with Pinus herrerae, which we can see in the next picture. On the left side, in the picture name A, we have a pure individual of Pinus Luz Maria, and we can see that it has not the best characteristics for the forest industry and the wood industry, because it is short, it is thin, it has a lot of branches on the trunk, so it's not very useful for the forestry or the wood industry. And on the right side, we have the hybrid of Pinus Luz Maria with gems of Pinus herrerae, and we can see that it is more useful for the industry because it is taller, is thicker, and doesn't have these branches all around the trunk, so we can make better wood and it can grow better in field. So we may think that hybrids have a better characteristic than pure individuals to survive and grow in field. And guess what? Will be completely wrong, because there is a high variability on vigor, robustness, and viability of the hybrid offspring. Actually, it is more common that hybrid offspring have less good characteristics than the pure individuals. This because different genetic barriers such as hybrid depression, hybrid inviability, or hybrid sterility, which affect the growth and capacity to survive in field. In this picture on the right side, we have two hybrids of Pinus engelmani. In the left side, we have a pure individual, and we can see that the needles are with a more vivid green color, maybe. They're like in order, but the hybrid is like more yellow color of the needles. We can see that it's like, doesn't come this day, you know, it's like a little messy. So it is important to do this kind of investigations, because the presence of hybrids in seed stands can influence the success of natural regeneration and reforestations. Additionally, with the analysis of the growth of hybrid and pure individuals, we are going to calculate the stomata density. Why? Because stomata density is an important tool that plants use to adapt to environmental conditions. These structures allow the entry of the carbon dioxide, which is necessary for photosynthesis, and limit moisture loss when the environmental conditions are unfavorable. Okay, we can see in the picture some difference between an open and closed stomata. The plants tend to close the stomata when the environmental conditions are not good for them. So the ability to close and open stomata depends and varies with the genetic factors. So we want to analyze, and we hope that there is a difference between stomata density in hybrid individuals and pure individuals. For this study, we have two different trials in the Sierra Madre Occidental. We can see it on the map painted in green. Sierra Madre Occidental is an important forest area in Mexico that is located in the northwest side of the country, and it crosses the state of Durango, which where I live, and this study was made. And in yellow, we can see Santiago Pasquiaro, which is the city where the trials are made in two different places. We can see it. One is called the Mesa Seca, and the other is the Mesa Alta. These kind of studies are known as reciprocal trials, and they are important. They are important because they allow us to measure the capacity and ability of the plants to grow under different conditions. Actually, even though that both sides for trials are close one to another, almost in the same area, they have different conditions. We have similar average temperature. We have 803 and 903 millimeters of annual precipitation. We got vegetation cover, which is lower in La Mesa Alta, as we can see in the picture. And there are grown up pine individuals in the Mesa Seca, which they are not in the Mesa Alta. There's say 100 meters of difference in altitude. And the predominant vegetation, as I said, is different. In Mesa Alta, we have like shrubs, small shrubs, and in Mesa Seca, we have tall pines around the area. What we did? Well, we evaluated the development of individuals in field. We assessed 5 of the most important wood species in Mexico, which are Pinus durangensis, Pinus teocote, which is more abundant actually in Mexico, Pinus engelmani, Pinus arizonica, and Pinus leiophylla. These individuals came from a previous investigation where they grow on the greenhouse conditions, and then we take it to the field to analyze how they adapt to these conditions. We measured the basal diameter with the vernier, digital vernier, and the height to the apical boot with the help of metric sand. Additionally, as I told you, we made an estimation of the stomata density in the needles. We collect 2 needles of the 3 most important species in field, the 3 more abundant. And we calculate like this. We count the number of lines present in both faces of the needle, adaxial face, which is like the top of the needle, and abaxial face, which is under the needle. We can see on the picture that the needle has lines of stomata all around, and we count these lines, and also we count the number of stomata that fit under a millimeter. We make a repetition of this with a second needle, and we calculate a mean stomata density to estimate the capacity of the ability of the plant to adapt under environmental conditions. Statistical analysis. Well, with all the data that we get, we made a post-cortical test, which help us to calculate the variation between significant differences between the growth in field in basal diameter and height to the apical boom. Well, after that, we make an spearman analysis because we want to assess the correlation between stomata density and growth parameters of basal diameter, height to the apical boot, and survival. We also estimate the variance of the survival thanks to a program which is called Delta, which compares the degree of differentiation between biological groups. Results, what we found in field. Well, after we analyzed basal diameter, we didn't find any significant differences between hybrid and pure individuals. The species with the bigger basal diameter was Pinus engelmani, but this is because it is how it grows. It first focuses on the basal diameter, and then it starts to grow in the height. But we actually found interesting data and significant differences in height to apical boot. When we compare together hybrid and pure individuals, we can see the hybrid individuals have a significant bigger height than the pure individuals. We also found significant differences in survival in 2 different species. One of them in Pinus arizonica, when hybrid individuals show a better survival in field than pure individuals, and in Pinus durangensis, which this process is repeated, we found a better survival on hybrid individuals. Stomata density, we didn't find any significant differences between hybrid and pure individuals of the same species, neither in the full comparison of hybrid and pure as a whole, but we found significant correlations between stomata density and growth parameters. We can see a positive significant correlation between stomata density and diameter, base diameter. We found another significant correlation, but in this time negative, with stomata density and both heights to the apical boot, and maximum height reached by the needles from the base to the end of the needle. And we found something interesting in the correlation between survival and stomata density. because hybrid individuals did have a significant correlation and positive with survival and stomata density, but not the pure ones. Apparently, the pure individuals have a decrease on the survival when the stomata density get to the highest point or the highest density. So in conclusion, what we found, no significant differences were found between basal diameter and height to the apical boot and maximum middle length between hybrid and pure individuals of the same species. But we did found significant differences when we compare the height to the apical boot in as a whole hybrids and pure individuals. Survival of hybrid was higher than pure hybrids. Survival of hybrids was higher than pure individuals in some species, as I told you, in Pinus arizonica and Pinus durangensis. As I told you, the hybrids as a whole show a greater height to the apical boot than the pure hybrids, and there is a correlation between stomata density and growth variables. So what we recommend? We recommend to continue this investigation, assess the difference between hybrid and pures around the years, and repeat this kind of investigation with different hybrids of different species, because there is a high viability in growth and adaptability of hybrid individuals, and natural hybridization is a natural force, and we all know that natural forces doesn't stop. Well, that's all for me. Thanks for your attention. I hope you have enjoyed this presentation, and of course I will attend every question.
Hello, everyone. Thank you for coming to my presentation today. My name is Garrett Evans, and I'm going to be telling you about my thesis research, which I have titled However, a fungal blight introduced in the early 1900s decimated the species, rendering it functionally extinct, meaning it's still present in its ecosystems, but serves no major role within them. However, researchers at SUNY ESF have used transgenic insertion to breaking down the harmful oxalic acid produced by the fungus, allowing the fungus and the tree to coexist with one another. We now have opportunities to reintroduce chestnut into its native ecosystem, which require very sound silvicultural methods. However, knowledge of the species, which is lacking in chestnut due to it being lost by the blight. In this case, we can use associate species as research analogs, such as oaks and hickories that have similar ecology and are believed to have filled the niche of chestnut left open by the blight. Northern red oak is often seen as the main analog for American chestnut and is regenerated using shelterwood systems, in which a portion of the overstory was removed, allowing light to the understory to initiate and promote regeneration. Once the new cohort is established, you remove the remaining overstory and release your new cohort. However, oaks are slow to increase in height after release, requiring competition control. This is an aspect of the species that is dissimilar from American chestnut that has been found to rapidly increase in height after release. This highlights one of the few differences between the species that may cause major differences in their silviculture. Another knowledge gap in American chestnut's ecology is its shade tolerance, which we can use to inform a species' preferred conditions and our management decisions. Oftentimes, groups of traits correlated with species of a certain shade tolerance go with one another. However, American chestnut is difficult to classify because it demonstrates traits from both ends of the shade tolerance spectrum. Persisting in the understory for long periods of time like a shade tolerant, while responding to increases in light with rapid height growth like a shade intolerant tree. One possible explanation for this combination of traits is a high phenotypic plasticity. Common example of this being the development of shade and sun leaves. This is when one genotype allows for individuals to acclimate and demonstrate different phenotypes in response to changes in their environment. Several studies have already confirmed that chestnut appears to be a highly plastic species. So the purpose of our experiment was to compare American chestnut to its associates under a gradient of available light. We did this in two ways. We looked at growth responses by quantifying seedling growth and comparing total size increases and changes with light, and looking at photosynthetic responses of seedlings by evaluating changes in leaf physiology and morphology. We hypothesized that chestnut would maintain faster growth rates across a light gradient as a result of higher phenotypic plasticity. And the applied goal of this experiment would be to evaluate the efficacy of shelterwoods in promoting chestnuts. Our study took place in Syracuse, New York, where we applied several cutting treatments to 2 forest blocks to create variation in light, both between and within treatments. Our first stand had an overstory of northern red oak, while our second stand had an overstory consisting of black walnut, sugar maple, and Norway spruce. So 2 very different research blocks. Our first treatment was a reference where we removed only invasive buckthorn. Our second treatment was a mid-story understory removal where the dominant stems were retained. Next, we did a thinning to 50% residual density, removing mid-story and understory, as well as a portion of the overstory. Finally, we installed several canopy gaps throughout the stand. Next, we planted chestnut in a grid across treatments and planted either an oak or hickory seedling nearby for comparison. We also chose to measure several naturally occurring sugar maple to include a shade-tolerant species in the study. We characterized the available light environment of each seedling using hemispherical canopy photos and expressed this using the gap light index as a percent between 0 and 100. You can see from this box plot, we were able to create a gradient of available light ranging from about 5 to 50% full sunlight. We quantified seedling growth by measuring the root collar diameter and stem length from the base of the stem to the tip of the terminal bud, taking these measurements in summer 2020 and 2021. We calculated the relative growth rate of both metrics in order to account for variation in the initial sizes of these seedlings. We used Lycor gas exchange systems to measure light response curves from a subset of our seedlings and fit these to a non-rectangular hyperbola. This calculates and extracts several parameters of interest, such as apparent quantum yield, which is how efficiently a leaf uses sunlight for photosynthesis, dark respiration rate, which is how much sucrose a leaf uses to maintain vital functions, and the light compensation point, telling us how much light is needed to achieve a net zero photosynthetic rate. We then took the sampled leaves and measured surface area, dry mass, and fresh mass. We sent them away for combustion analysis to determine percent nitrogen and use these metrics to calculate leaf mass per area, mass-based nitrogen, and area-based nitrogen. These are all morphological traits of leaves that are highly correlated with photosynthesis and available light. We then did a linear regression to determine the correlation of our response variables and a light gradient, doing a backwards stepwise model selection to determine what other variables increase the fit of our model. We always kept an interaction between species and GLI, as this is critical for testing our hypothesis of variation between species across light. And we required 10 living individuals to be included for our growth analysis. So we now take a look at some of our results. Here we have relative growth rate results. You can see the relative growth rate of stem length on the y-axis and GLI on the x. At low light, there is comparable growth between all species. However, as GLI increases, you see the RGR chestnut in red increasing much more than other species. These others showed very little change across our light gradient. Similarly, when we look at the root collar diameter growth rate, chestnut increases much more than any other species and having much higher growth at high light. However, we do see that shade-tolerant sugar maple in purple has a similar increase to chestnut. But we see red oak in blue and pignut hickory in green showed very little change in relative growth rate, both in stem length and root collar diameter, as GLI increased. We also looked at these metrics as percent cover increased and found that percent cover had little variation and effect on relative growth rate. So the main takeaway of our relative growth rate results being that chestnut had the greatest response across a GLI gradient. Next, we have our photosynthetic traits with max photosynthetic rate on the Y. Here, we compared 20 chestnut, 20 red oak, pignut hickory, and sugar maple across our GLI gradient. There was no statistical variation between species or across GLI for maximum photosynthetic rate, which was a bit unexpected. We did see some variation in apparent quantum yield, showing that chestnut leaves become more efficient with light, sugar maple leaves, a shade tolerant, becoming less efficient as light increases, and red oak and pignut hickory remaining about the same. We did see significant increases in dark respiration as light increased. However, this was the same across species, meaning that as light increased, leaves were using more energy to maintain vital functions. Similarly to our dark respiration results, we see similar light compensation point results. Because if leaves are using more energy at higher light, they're going to have to produce more sugars in order to break even. These results were a bit unexpected based on our growth results, as we didn't see variation in maximum photosynthetic rates in the same way we expected. And our dark respiration and light compensation point results show that changes were similar across species. Next, we have the results of our leaf trait analysis, showing that LMA on the y-axis varied between species, but did not vary across GLI, with red oak having the highest LMA, followed by the others. Area-based nitrogen, oak had the highest, followed by chestnut, hickory, and maple, but again, no change across a GLI gradient. Finally, we have the results of our mass-based nitrogen analysis, showing that there was some significant changes across GLI, with chestnut and red oak decreasing and sugar maple increasing. But overall, these nitrogen results and LMA tell us that there is comparably low leaf morphological plasticity across species and across a GLI gradient. So the conclusions of this study are that American chestnut had the greatest stem growth at high light and the greatest increases in RGR, telling us it has a higher growth plasticity. However, we saw little variation in the way of leaf physiology and morphology, suggesting low leaf plasticity in response to light. These results were a bit unexpected, as we thought that the higher growth of chestnut would be explained by increases in photosynthesis and respiration, but this wasn't the case. It appears that chestnut's growth plasticity is not driven by photosynthetic changes. Instead, a possible explanation is that variation in growth is due to difference in seedling growth habits. It's commonly been found that northern red oak seedlings allocate the majority of biomass early on towards a root system, allowing them to resprout quickly after disturbances, while other studies show that chestnut allocates early biomass towards shoot growth, increasing in height after increases in light. However, what we can take from this study is that chestnut grows well under high light environments, and the results show that shelterwood systems may be a viable method for reintroduction of American chestnut back into portions of its native range. And with that, we will now be taking any questions you may have. Thank you for your time.
Hello, good afternoon. My name is Carlos Adrián Vázquez Berumen. I am a student of the doctorate program in Agriculture and Forestry Science and the Universidad Juárez del Estado de Durango. The title of my work is Development of Putative Hybridous and Poor Individuals of 5 species of the genus Pinus in Durango, Mexico. We have you a brief introduction to the topic, which that use that natural hybridization refers to all crossbreeding that occurs between genetically distinct individuals. With this process, different problems can occur, such as low adaptability and mean that the species do not develop in adequate way, infertility, and loss of their reproductive characteristics. For instance, the research work consists of evaluation, the development of two trials of hybrids and food samples of Pinus arizonica, Pinus dorangensis, Pinus saint-germani, Pinus leiophylla, and Pinus tecote implantation. This investigation will connect user information to make decisions on the potential use of hybrids for future reforestation. This research is a follow-up to several studies in the year 2015 in the research discriminations of set stand of various Mexican pine species based en 43 dendrometric, prismatic, edaphic, and genetic traits. In this study, the parent tree were evaluated and set was collected from the study samples. In the other two studies, set germination was evaluation and seed sowing was performed. The main objective is evaluate the development interpretation of protective and poor hybrids of Pinus dorangensis, Pinus arizonica, Pinus saint-germani, Pinus tecote, and Pinus leiophylla. The particular objective is determine the difference in diameter and average height between hybrid and poor centuries of 5 species of the genus Pinus by comparing the development of the centuries in the fruit in relation at the soil and climatic conditions of the set stand where the parent trees were grown. The new hypothesis there will be no significant difference in the development of hybrid and forest standards of the 5 Pinus species in relation to the environmental conditions of the stand where the parent trees were grown. The following figures show the two trials of the studies are located in Umafor, 1005 in Santiago Papasquiaro, one calle Mesa Alta, and the other Mesa Seca. As mentioned, this work that is being followed and worked at the same time. That is why we are using the same images as partner Ricardo. Condition of the features. 25 edaphological variables and 18 bioclimáticas variables used by Hernández Velasco et al. 2021 were taken. Some of these are calcium, magnesium, sodium, mean annual temperature, winter precipitation, minimum temperature in the coldest month, maximum temperature in the hottest month. In the table 1, variables of the stand where the parent trees green. And materials y métodos GPS was used to locate the seeds, and aluminum platters were used to mark the individuals and identify them by space, place of origin, number, and whether they were poor or hybrid. For measuring the basal diameter, we used a digital vernier 2 mm. Where a tape measure was used to measure the height. We perform static analysis that were performed Shapiro-Wills analysis, Kruskal-Wallis non-parametric text, Spearman non-collinear text less than 0.70, and EWG and variables edapological and bioclimatical and experiment correlation tests. The Shapiro will text showed that the data of the study did not present normality. No significant differentials were found in the crystal Wallis analysis for diameter and height. In the figure 2 showed the results of experiment correlation of diameter and height with edapological and bioclimatical variables. I can be observed that in the hybrid individuals there is correlation with studio variables such pH, mean, yield, and wind, and variables edapological, potassium, y phosphorus, and individuals hybrid. In the next figure showed the relationship between mean height and average and full standards in the 46 months after planting and soil potassium concentration in the stems of their parent trees. With the alternate result, I can be knocked down. Average standards show a relation between mean diameter with mean and pH in contrast to current standards. In height, there was a correlation with potassium and phosphorus in every standards, while in the wind variable there was a positive correlation in both average and poor standards. With this, I can be concluded that every standard have a better correlation with edapological and bioclimatic variables. It is recommended to follow up on this research project to see how it develops over time. Some of the literature cited. With this, I finish my presentation. Thank you for your attention